Of the schedule for this evening. We're going to go through introductions / public relations. Jayani will give her presentation. We'll have a brief Q&A and then a quick poll before we let you go. A quick introduction to the American Chemical Society. It is the world's largest scientific community. Members have access to career development and networking, member groups and communities, as well as conferences and events, and have opportunities to earn funding and awards. Oh, let me make sure I get it all on here. There we go. All right, so as part of ACS career development, members have access to the CNE and jobs database, free career counseling, career pathway workshops, and Chem IDP. For those that are interested and have a active membership, you can access virtual office hours where you'll be connected with a career counselor via Zoom. They are held the first Thursday of every month from 12 to 1:30. You can find that link here. The next one will be on December 1st. ACS members also have access to the Women Chemists Committee, Minority Affairs Committee, and the Younger Chemists Committee. That's us. Our vision is younger leaders transforming the world through chemistry, which means that we advocate for, develop, and support rising chemists like you. The Eastern US YCC Partnership is a subsection of the National YCC. We have several local section YCCs that participate, including the Northern and Eastern New York sections, Connecticut Valley and Philadelphia, Virginia, St. Louis, Puerto Rico. And we also have some international groups as well, including the Universidade Federal de Goiás from Brazil. We've hosted several day in the life events over the past 2-3 years, which has earned us 2 Chem Luminaries for the day in the life. In 2021, we earned Best Activity or Program Stimulating Member Involvement. And this year, we won Outstanding Continuing Public Relations or Communications Program of a local section. If you'd be interested in getting involved, you can join or start your own local section YCC, attend YCC events at ACS national meetings, or apply for YCC sponsored awards. At ACS conferences and events, let's see, one more link for you guys. There we go. At ACS conferences and events, there are several that you can choose from. We have ACS national meetings, regional meetings, as well as green chemistry conferences. The next ACS national meeting is going to be in spring. I believe it's the end of March. It's going to be in Indianapolis, Indiana, and that will be hybrid. Oh, there we go. All right, I will pass it over to my co-host, Selena Kunzig, to introduce our speaker today.
Thank you, Ashley. So as Ashley said, I'm one of the co-hosts. My name is Selena Kunzig, and I have the pleasure of introducing Dr. Jayani A today. They recently graduated from VCU with their PhD in chemistry in 2022, and they're currently a Postdoctoral Research Fellow in the Department of Biomedical Engineering at Duke University. Their current projects involve designing and engineering proteins for targeted peptide degradation and developing screens for identifying protein drug targets. Dr. Christopher, the floor is all yours.
And as a final note, if you have questions during the presentation, please feel free to pop that in the chat. We'll have an open Q&A where you can unmute yourself after Dr. Christopher is finished speaking. There you go.
Okay, let me share my screen. Okay. Okay, thank you, Selena and Ashley, for the introduction. And my name is Jayani Christopher. I'm currently a postdoctoral associate in Duke Biomedical Engineering. I am very excited to be here sharing my experience as a protein biochemist. So today, basically, I will be telling you how my day looks like in my new role, basically. So moving on, this is the outline of my presentation. First, I will be giving a brief background about myself, my education, and where I came from and all that. Then I will be discussing about my PhD research and also about my current postdoc research. And then a little bit of what I do on a daily basis right now. And then also some lessons I learned, which could be I think which could be helpful to whoever is following the same path. So with that, moving on to the background. So before everything, where did I come from? I'm originally from Sri Lanka, which is a small island right next to India. I think for a lot of people who are in academia, they are familiar with what Sri Lanka is, because in academia you find like a lot of people from our country. Probably some of them might be here. I saw like some people in the meeting. So even though we are a small island, we are like very diverse geographically. There are so many beautiful beaches and mountains. So if you ever visit, make sure you explore them. To be specific, I am from the central part of Sri Lanka, which is a small city in mountains called Kandy. We have like Kandy is a city with a lot of historical significance. So make sure to visit Kandy if you ever visit Sri Lanka. With that, my educational background, I did my bachelor's degree in University of Peradeniya, Sri Lanka, which is basically ranked #1 currently in Sri Lanka. So I majored in chemistry and graduated in 2016. So after that, I came to USA in 2017 to do my PhD, and I graduated from VCU in 2022 with my PhD in chemistry. And after that, in July 2022, I joined Duke Biomedical Engineering as a postdoc. So that's where I am right now. And then this is a little bit about me outside of science, what I do when I don't do science. I love cooking. These are some of the dishes that I prepared, and I love taking pictures of them and posting that on Instagram. And I love feeding people, which I don't get to do much ever since I moved to like the new city. But I love cooking Sri Lankan food. I'm also like learning about like other cuisines as well. And also another thing that I love doing is volunteering and networking, which has been like really helpful for me to meet people and also to build my career. These are like few events that I attended after I moved to like the Duke University. These are like some events organized by Duke Postdoc Association. And also I love traveling when I get time. I like exploring. I like going to places, meeting people, and like, trying out new food. Oh, that's pretty much about me when I don't do science. With that, I'm going to move on to the science part of my presentation. So I did my PhD research in Dr. Ashton Kropp's lab in VCU chemistry. My research was designing biosensors for macrolide antibiotics, and basically, I'm going to like talk about my research very briefly today because of the time constraint. I will be like talking about like one of the projects. So I was working on improving the sensitivity of this macrolide antibiotic biosensor. And also I developed a method as a growth selection for macrolides, and also worked on developing ratiometric biosensors for simultaneous detection of 2 macrolides. Other than that, there was like another broader project I worked on, like protein engineering, basically where I incorporated unnatural amino acids to histone proteins and other proteins as well, which recently got accepted for publication. So I'm excited about that. So moving on to my research project. So I will be giving a little bit of introduction here. So my entire research was based on macrolides. Macrolides are a class of antibiotics characterized with the macrolactone ring. For an example, like erythromycin. These kind of compounds are biosynthesized by large assembly line enzymes known as polypeptide synthases. They have several enzyme modules, and these enzymes are really, really complex. One thing we know about antibiotics is bacteria can get resistant to existing antibiotics. In that case, we always need novel antibiotics. So if we are synthesizing novel macrolides, combinatorial biosynthesis is a good strategy, where basically you can swap the modules, the enzyme modules in polypeptide synthesis and design a new compound. However, it's a bit, it's challenging because polypeptide synthesis are large and also very complex. So it's difficult to work with them. On the other hand, to do late stage modifications for these compounds, there are no tailoring enzymes available. So in that case, Crop Lab came up with an alternative strategy, which is known as directed evolution, which is basically mimicking the process of natural selection. So basically, what do we do here? If we want to modify a macrolide and introduce a modification, a target modification like in here, it can be done enzymatically. But however, it's not always there is an enzyme available to do such specific modifications. So in that case, we can mutate an existing enzyme and create a library of enzyme variants and screen that library in order to identify an enzyme which is doing the target modification. However, when we are using macrolides for this screening, it's again challenging because macrolides are not chromophores or fluorophores. So there is no way to detect that. In that case, it's important to have good screening methods or high throughput screening methods. So that's where my research came into play. I developed biosensors for screening these kind of libraries. So the biosensor that I'm going to talk about today is developed from this gene cassette here, which was found from an erythromycin-resistant E. coli variant. It has a promoter sequence, and downstream of that, there are 3 genes, MPHA, MRX, and MPHR. All 3 of them are expressing important proteins. What happens here is MPHA is expressing a kinase which can phosphorylate erythromycin at the disosamine sugar. And then the second gene is MRX. It expresses a membrane protein that can eliminate the antibiotic from the cell. And then we have MPHR, which is responsible for this whole process, which is a transcriptional repressor. What basically MPHR does, MPHR can bind to the promoter sequence upstream of this gene cassette, and it repress the transcription. So these genes will no longer can express the protein. And then in the presence of erythromycin, MPHR will undergo a conformational change and release from the promoter, and it will de-repress the transcription, and so that the subsequent proteins can be expressed. That is the basic concept of concept that we use to develop the biosensor. So this is the biosensor that has been used in crop lab. I'm sure still this is that they're using it. Our biosensor consists of 2 plasmids. Basically, the genes from the original gene cassette was put into 2 plasmids here. The first plasmid has the MPHR, and this is the promoter sequence to which MPHR protein can bind. Other than that, we introduced this GFP reporter gene, which can express the green fluorescence protein. And also the second plasmid contains the MPHA, which is expressing the kinase to phosphorylate erythromycin. This was introduced with the hypothesis that erythromycin phosphate is a better ligand for MPHR compared to erythromycin. Later, this was confirmed by one of my colleagues from crop lab. So basically the same concept here. In the presence of erythromycin, MPHR would undergo the conformational change released from the promoter, so that will de-repress the transcription of GFP, giving green fluorescence. So only in the presence of erythromycin, there will be green fluorescence. My role, my goal for the first project was to improve the sensitivity of this existing biosensor. So for that, I used an approach known as gene knockouts. Basically, it is disruption and inactivation of chromosomal genes so that the respective proteins cannot get expressed. Basically, I worked with 2 types of knockout strains. The first one is a single gene knockout strain, which is associated with the ACR-AB Tulsi multi-drug efflux pump. What efflux pumps do, they can eliminate antibiotics from the cell. So if we knock out the Tulsi gene, the Tulsi protein cannot be expressed. So that efflux pump will be dysfunctional, and then the elimination of the antibiotic will be stopped. So if we transform our biosensor plasmid system inside these cells, inside these knockout strain, there will be more antibiotic inside the cell to be detected by the biosensor. So our hypothesis was when there's a gene knockout, that will increase the sensitivity of the biosensor. Second type of strain is a double gene knockout. It's a combination of this Tulsi knockout as well as the second knockout is a Bambi knockout. Bambi is a protein from this beta barrel assembly machinery complex. This is responsible for folding and insertion of outer membrane proteins to the membrane so that the membrane becomes impermeable to macrolides. So when the Bambi gene is knocked out, this BAM complex will not be functional. It cannot introduce the outer membrane protein, so that membrane becomes more permeable for macrolides, and there will be more influx. So with the double knockout strain, there will be more influx as well as there is no efflux. So there will be more antibiotics within the cell to be detected by the biosensor, and we expected higher sensitivity with the double knockout strain. So to study that, I conducted some bioassays. The first one is called an agar diffusion assay. So how do you do it? First, you transform the biosensor plasmids in E. coli, the wild type, or the knockout strains, and then out of that, pick colonies and grow a liquid culture, and then use that culture to plate on an agar plate, and then spot the plate with filter paper discs containing different concentrations of erythromycin, and then imaging the plates. So these are my plate images. With the wild type strain, the green color is due to the green GFP fluorescence. So with the wild type strain, I was able to see fluorescence down to 10 micrograms. When it comes to the single knockout strain, the fluorescence was seen down to 1 microgram. There's like a little bit of fluorescence here. It's like barely visible, but it was there. And then when it comes to the double knockout strain, again, the fluorescence was seen down to 1 microgram, but it was more fluorescence compared to the single knockout strain, proving our hypothesis was correct with in here. The second type of assay I conducted was a quantitative assay. Basically, here the liquid culture was used to grow a 96 well plate with with different concentrations of erythromycin, and then this 96 well plate cultures were grown, and I used those cultures to measure fluorescence using a fluorescence plate reader, and then finally plotted the fluorescence response against erythromycin concentration. So according to this curve, compared to the wild type, the knockout strains definitely had like very high fluorescence. And however, when it comes to when we compare the single knockout strain to double knockout strain, I saw a decline in the fluorescence of the double knockout strain that is due to the cell death caused by high concentrations of erythromycin retaining in the double knockout strain, which also corresponds to this halo effect or the zones of inhibitions in these agar plates. So basically in this project with the knockout strategy, I was able to improve the sensitivity of this biosensor. So this is about like one of the projects I conducted in the grad school. There's a lot more I can talk about, but I don't think I have time for that. So with that, I'm moving on to my postdoc research. So I currently work with Dr. Pranam Chatterjee in Duke Biomedical Engineering. We are called, our lab is called Programmable Biology Group, which is because we have two separate groups. One is a computational group and the second is an experimental group. So obviously I belong in the experimental group, and this is like a fun picture my group members took during Halloween when they became minions. Unfortunately, I could not be there in this picture because I got COVID during the same time. But yes, so in our programmable biology group, we do three things basically. First is genome editing, second is proteome editing, and third is cell engineering. My role is mostly leading the projects in proteome engineering. So little bit about my project. I work on designing peptide-guided degradation platform as therapeutics. Here, what we do is called targeted peptide degradation. What basically happens is there are disease-causing proteins that can be degraded using the ubiquitin proteasome system. Like that could be degraded by hijacking the ubiquitin proteasome system by linking this the disease-causing protein with an E3 ligase. However, to link that, we need a linker. So in that case, our lab uses this technology known as ubicobody, where we combine where we replace the substrate binding domain of E3 ligases with a peptide, and that peptide is also able to bind to the target protein. Then when the E3 ligase is bound to the target protein, the target protein is degraded. This can be an approach to develop protein therapeutics. So basically, we have an amazing computational group who use protein language models to develop like these peptide linkers. And my work is basically testing or like engineering these peptides and developing assays to screen them. So that is basically what I do in my research project. With that, moving on to like what I do on a day. So there are so many things that I do. Like I spend most of my time in the lab, sometimes like 12 to 15 hours, even it could be more than that. Like it's sometimes like it's a lot of work, but I like doing what I do. So basically, first thing I do, as a researcher, I conduct experiments, and then something all of us should do is literature search and stay up to date with the current literature. And then I use that knowledge to design experiments, which is sort of like a new experience to me after moving to the postdoc, because in grad school, mostly like you get to do what your PI tells you to do. And then I also work on writing manuscripts and grants. And one of the most significant role that I have in the lab is like managing the lab. I basically joined a lab which was freshly started in the university. So I had to kind of, learn all the logistics, how to order things, or how to manage the safety or the waste and all sorts of things. So I'm currently like doing it's part of my job as well. And the most rewarding part, I would say, mentoring my students. I have a bunch of amazing, really smart students whom I love to mentor, and I really enjoy doing that. I do have like undergrads, master students, and grad students who are like really like extremely hardworking and like very, very helpful to me. And also, since we are a new lab, I work on implementing new protocols to the lab. Always, our lab prefers like following a standard protocol for everything to maintain the consistency of the research. So this is basically what I do on a daily basis. With that, I want to share some of, based on my experience as a researcher or a biochemist, what I learned throughout this process. The first thing is, always to like be resilient. It's not like, everything is going to work all the time. Whoever is like doing research, I know like most of the people who are in here would agree with me. It's like 90% of the time you're going to fail, but you have to really be patient and keep working and believe that it's going to work. And secondly, believe in yourself. It's something that I always keep in my mind. I came from a small country, and managed to come this far and to be in like one of the really big universities in USA. So I just always believed that I was able to do it. And another thing, taking up challenges. I did like, several transitions. I did not get to present about my undergraduate work. Like my undergraduate work was very different from my grad school experience, and also moving from chemistry to biomedical engineering again was like challenging. So always open to be take challenges, and also not to be afraid to try new fields. It's like good to learn different areas of science. And also another important thing, always be open to learn from others, or like from literature, from other people. It could be your mentor, it could be your friend, colleague, or it could be someone senior to you, junior to you. Sometimes I learn things from my students too. So which is something I always keep continuing, and always, and also most important thing, love what you do. Like just be passionate about it. Otherwise, it's like it's gonna get stressful. But if you don't like it, it's gonna be really hard. And at the end, as scientists, always we will have to work hard no matter what, like whether you are in academia, industry, it doesn't matter, you will have to work hard. So keep pushing yourself until you reach your goal. With that, I would like to acknowledge ACS-YCC for giving me the opportunity. I'm honored to be here. Thank you for inviting me to do to be a part of the Day in a Life series. And VCU for giving me the opportunity to pursue my education in USA. By far, out of all the universities I've been in, VCU is my favorite. And then Duke University for giving me the opportunity to do the work that I do right now. And NIH for funding my research, Dr. Pranam Chatterjee, my current PI, and all my lab members who has been like really amazing. And I enjoy working with them, and especially Dr. Kropp from VCU, who has been an amazing mentor, and he has been like, I learned so much from him. Like most of the things I do today, I learned from Kropp Lab. And I want to thank all the lab members, Dr. Kropp, who's like still in touch with me, which is really amazing. And especially all my friends and family, big shout out to my friends from Richmond. Without them, things would like nothing would have been possible. So, and thank you all for joining in, and I'm happy to take questions. I have got like a lot of questions previously. I might have answered some of them. I don't know how this should work now. Ashley, like if you can like tell me how I should do about it.
Yeah, thanks so much, Jayani. So Selena is going to lead our Q&A tonight. So I'm going to go ahead and pass it over to her. You can unmute yourself, ask questions.